Welcome to Cosmic Whispers, where cosmic mysteries come alive. Your journey beyond the stars begins now. Beneath the towering canopies of Earth's tropical rainforests lies a secret universe that most humans will never witness. While explorers map distant galaxies and probe the deepest ocean trenches, an entire realm of life thrives just meters below. The jungle floor, a world so alien it might as well exist on another planet. These subterranean jungle ecosystems represent one of Earth's most enigmatic frontiers. Unlike the familiar caves found in temperate regions, tropical underground systems exist in a state of perpetual biological flux. Here, the boundary between surface and subsurface dissolves into a gradient of interconnected life forms, each adapted to conditions that would challenge our understanding of what constitutes a livable environment. The story begins with water. In tropical regions, intense rainfall creates unique hydrological conditions that carve elaborate networks of underground chambers, tunnels, and waterways. Unlike arid cave systems, formed primarily through chemical weathering over millions of years, jungle cave networks develop through a combination of rapid water flow, root penetration, and the constant biochemical activity of countless microorganisms. Temperature regulation becomes crucial in these environments. While surface temperatures in tropical jungles can fluctuate dramatically between day and night, subterranean chambers maintain remarkably stable conditions, typically ranging between 24 and 28 degrees Celsius year-round. This thermal stability creates microclimates that support species found nowhere else on Earth. The absence of sunlight might suggest these environments would be biologically barren, but recent scientific expeditions have revealed the opposite. These underground ecosystems teem with life forms that have evolved extraordinary adaptations. Many species exhibit complete loss of pigmentation, enlarged sensory organs, and metabolic processes that operate on entirely different principles than their surface-dwelling relatives. Every great journey starts with a few brave explorers. Right now, Cosmic Whispers is just beginning, and your support means everything. If you enjoy unraveling the mysteries of the universe, hit subscribe. It's free, and it helps us grow this community together. Scientists estimate that less than 5% of these subterranean jungle ecosystems have been systematically studied. Each expedition reveals new species, from translucent fish that navigate by detecting minute changes in water pressure to arthropods that have developed echolocation, capabilities rivaling those of bats. These discoveries challenge fundamental assumptions about evolutionary biology and the limits of life on Earth. The complexity of these systems extends beyond individual species. Entire food webs operate in perpetual darkness, sustained by organic matter that filters down from the surface world above. Decomposing plant material, carried by underground streams, becomes the foundation for elaborate nutrient cycles that support hundreds of interconnected species. Recent technological advances have revolutionized our ability to study these environments. Specialized underwater cameras, environmental DNA sampling techniques, and remote sensing equipment allow researchers to document life forms without disturbing their delicate habitats. These tools reveal ecosystems operating under principles that seem to defy conventional ecological theory. Perhaps most remarkably, many subterranean jungle species demonstrate evolutionary adaptations that occurred independently across different continents. This convergent evolution suggests that underground tropical environments create selective pressures so specific that similar solutions emerge regardless of geographic isolation. The implications extend far beyond academic curiosity. These ecosystems may hold keys to understanding how life might exist in extreme environments on other planets. The metabolic pathways discovered in jungle cave organisms could inform biotechnology applications, while their unique biochemistry might yield new pharmaceutical compounds. As we prepare to explore these hidden worlds in greater detail, we stand at the threshold of discoveries that could reshape our understanding of life itself. The creation of subterranean jungle ecosystems begins millions of years before the first organism ventures into darkness. Unlike the limestone caves commonly found in temperate regions, tropical underground systems form through complex geological processes that operate under entirely different conditions. In equatorial regions, intense seasonal rainfall creates hydrological forces unmatched anywhere else on Earth. Annual precipitation often exceeds 4,000 millimeters, with individual storms delivering more water in hours than temperate regions receive in months. This relentless hydraulic pressure carves underground networks with remarkable speed, geological timescales compressed into millennia rather than millions of years. The bedrock composition in tropical regions plays a crucial role in cave formation. Many jungle areas sit atop ancient sedimentary formations, rich in soluble minerals. However, unlike the gradual chemical weathering that creates temperate caves, tropical cave formation involves aggressive biological and chemical processes working simultaneously. Root systems from towering jungle trees penetrate deep into the earth, sometimes reaching depths of 30 meters or more. These roots don't simply grow around obstacles. They actively secrete organic acids that dissolve rock formations. The biochemical weathering caused by root exudates can carve passages large enough for human exploration within centuries. Temperature. Differentials create unique geological stresses in tropical underground systems. Surface temperatures can reach 40 degrees Celsius during peak daylight hours, while underground chambers maintain steady temperatures around 26 degrees Celsius. This thermal gradient causes repeated expansion and contraction cycles that fracture bedrock in distinctive patterns. Water chemistry in tropical underground systems differs dramatically from temperate cave environments. High levels of dissolved organic compounds carried by surface runoff through dense vegetation create acidic conditions that accelerate rock dissolution. These organic acids, primarily humic and fulvic compounds, can achieve pH levels as low as 4.5, acidic enough to dissolve significant amounts of limestone and other carbonate rocks annually. The rapid formation rate of tropical cave systems creates geological features rarely seen elsewhere. Flowstone formations develop within decades rather than millennia. Speleothems, the technical term for cave formations like stalactites and stalagmites, grow at rates up to 50 millimeters per year in tropical conditions, compared to typical growth rates of less than 1 millimeter per year in temperate caves. Underground river systems in tropical regions often maintain year-round flow, fed by consistent rainfall and elevated water tables. These streams carve elaborate channel networks that can extend for kilometers underground. Some tropical cave rivers emerge from underground systems with flow rates exceeding 10,000 liters per minute. Sediment transport in these underground rivers creates unique depositional environments. Fine clay particles rich in organic compounds from surface vegetation accumulate in quiet pools and backwater areas. These sediment deposits become crucial habitat zones for specialized organisms adapted to life in complete darkness. The structural stability of tropical cave systems presents unique challenges. Rapid formation processes often create chambers with thin roof sections that require constant monitoring by researchers. Some documented cave chambers span areas larger than football fields while maintaining roof thickness of less than 2 meters. Mineral precipitation patterns in tropical caves reflect the unique chemistry of their formation environment. Gypsum crystals, typically rare in temperate caves, form elaborate displays in tropical systems due to high sulfate concentrations in groundwater. These crystal formations create microhabitats that support highly specialized bacterial communities. Hydrothermal activity occasionally influences tropical cave development, particularly in regions with recent volcanic activity. Warm groundwater, heated by geothermal sources, can maintain underground chambers at temperatures exceeding 35 degrees Celsius, creating tropical greenhouse conditions underground. The geological foundation of these systems continues, evolving even as biological communities establish themselves. Active speleogenesis, the technical term for ongoing cave formation, means that tropical underground ecosystems exist in a state of constant physical change, with new passages opening, while others become sealed by mineral precipitation or structural collapse. In the perpetual darkness of subterranean jungle ecosystems, life begins with organisms invisible to the naked eye. While surface jungle communities depend on photosynthesis to convert sunlight into biological energy, underground systems rely on microscopic pioneers that have mastered entirely different strategies for survival. Bacterial communities in tropical cave systems exhibit diversity that rivals any ecosystem on Earth. Recent molecular analysis techniques, particularly environmental DNA sequencing, have revealed that a single cubic centimeter of cave sediment can contain over 10,000 distinct bacterial species. This microbial diversity exceeds that found in most surface soil environments, despite the apparent uniformity of cave conditions. Chemosynthetic bacteria form the foundation of these underground food webs. Unlike photosynthetic organisms that harness light energy, chemosynthetic bacteria derive energy from chemical reactions involving compounds like hydrogen sulfide, methane, and various nitrogen compounds. These chemical energy sources arrive through groundwater flow, organic matter decomposition, and mineral weathering processes. Sulfur-oxidizing bacteria represent one of the most important primary producer groups in tropical cave ecosystems. These organisms convert hydrogen sulfide, naturally produced through anaerobic decomposition of organic matter, into sulfuric acid while capturing the released energy for biological processes. The acidic byproducts of this metabolism contribute to ongoing cave formation by dissolving surrounding rock. Methanotrophic bacteria utilize methane seeping from deep geological formations or produced by other microorganisms. These bacteria can achieve remarkable population densities, up to 1 billion cells per gram of sediment, in areas where methane concentrations remain consistently elevated. Their metabolic activity produces organic compounds that support entire communities of secondary consumers. Iron-oxidizing bacteria thrive in tropical cave environments where groundwater contains elevated ferrous iron concentrations. These organisms create distinctive rusty-orange biofilms on cave walls and underwater surfaces. The oxidation process generates both energy and unique chemical environments that support additional microbial species. Nitrite-oxidizing bacteria complete crucial nitrogen cycling processes in cave ecosystems. These organisms convert ammonia, compounds derived from decomposing organic matter, into nitrites and nitrates that serve as essential nutrients for other community members. This nitrogen transformation process enables complex food webs to develop in environments with limited external nutrient input. Biofilm formation creates complex three-dimensional habitats within cave systems. Bacterial communities secrete extracellular, polymeric substances that form sticky matrices, allowing multiple species to coexist in intimate proximity. These biofilms can cover vast areas of cave surfaces, creating what researchers describe as living wallpaper that supports higher-level organisms. Extremophile bacteria push the boundaries of life in cave environments. Some species tolerate pH levels below 3.0, while others thrive in temperatures approaching 40 degrees Celsius near geothermal features. These extreme adaptations provide insights into how life might exist in similarly harsh environments on other planets. Symbiotic relationships between different bacterial species create metabolic networks of remarkable complexity. Some bacteria produce waste compounds that serve as energy sources for other species, creating closed-loop systems that maximize efficiency of limited resources. These partnerships can involve 5 or more bacterial species functioning as integrated units. Seasonal variations in bacterial activity reflect changes in surface conditions above cave systems. During dry seasons, reduced water flow concentrates nutrients and creates different selective pressures than wet season conditions. Some bacterial species enter dormant states during unfavorable periods, only to resume active metabolism when conditions improve. Recent discoveries have revealed bacteria capable of surviving on extremely low energy budgets, metabolic rates, up to 1,000 times slower than typical surface bacteria. These organisms challenge fundamental assumptions about the minimum energy requirements for life and suggest that similar organisms might exist in subsurface environments throughout the solar system. Horizontal gene transfer between cave bacteria occurs at rates higher than in most surface environments. The close proximity of different species in biofilm communities facilitates exchange of genetic material, allowing rapid adaptation to changing environmental conditions and the evolution of novel metabolic capabilities. The collective biomass of bacterial communities in large tropical cave systems can exceed several tons of living material, making these microscopic organisms among the most significant biological components of underground ecosystems. Evolution has produced some of its most remarkable innovations in the eternal darkness of subterranean jungle ecosystems. Animals that have colonized these environments have undergone profound physiological and behavioral transformations that seem to rewrite the rules of biology. Troglobiotic species, organisms that spend their entire life cycles underground, represent evolution's most extreme responses to cave conditions. Unlike temporary cave visitors that retain connections to surface environments, true cave dwellers have severed all ties with the sunlit world above, developing adaptations so specialized that return to surface conditions would prove fatal. Vision. Loss represents the most obvious adaptation to perpetual darkness, but the evolutionary pathways involved reveal fascinating complexity. Rather than simply losing functional eyes, many cave species have repurposed the developmental energy normally invested in visual systems. The Mexican tetrafish, found in underwater cave systems throughout Central America, demonstrates this principle perfectly. Cave populations have not only lost functional eyes, but have developed enlarged lateral line systems that detect minute water movements with extraordinary precision. Sensory enhancement extends far beyond improved mechanoreception. Cave-dwelling arthropods have evolved antennae that can exceed twice their body length, covered with chemoreceptors capable of detecting individual molecules in the surrounding environment. These chemical sensing capabilities allow navigation through complete darkness with accuracy that rivals visual navigation in surface environments. Metabolic adaptations in cave animals challenge conventional understanding of energy requirements. Many troglobiotic species have reduced their metabolic rates by 30 to 50% compared to surface relatives. This metabolic suppression allows survival during extended periods of food scarcity. Some cave fish can survive over 2 years without feeding, living entirely off stored body fat. Reproductive strategies in cave environments have evolved remarkable flexibility. Many species have abandoned seasonal breeding cycles, instead reproducing opportunistically when resources become available. Some cave fish produce fewer but larger eggs, investing more energy per offspring to increase survival chances in resource-limited environments. Pigmentation loss in cave animals results from more than just lack of selective pressure for coloration. The biochemical pathways that produce pigments require significant metabolic investment. Cave species redirect this energy toward other systems, often developing translucent body walls that allow internal organs to be visible, a feature that provides researchers with unique opportunities to study living physiology. Thermal regulation in cave animals reflects the stable temperature conditions of underground environments. Many species have lost the ability to regulate body temperature across wide ranges, instead becoming precisely adapted to the narrow temperature variations found in their specific cave systems. This specialization makes them extremely vulnerable to climate changes that alter underground temperatures. Behavioral adaptations in cave ecosystems include development of sophisticated spatial memory systems. Without visual landmarks, cave animals must navigate using chemical trails, tactile cues, and mental maps of extraordinary complexity. Some cave-dwelling salamanders can navigate directly to specific locations across hundreds of meters of underground terrain. Social behaviors have evolved unique characteristics in cave environments. Some species that live solitarily on the surface form complex social groups underground, perhaps as an adaptation for information sharing about resource locations. Cave-dwelling bats, for example, often form larger colonies underground than their surface roosting relatives. Longevity increases dramatically in many cave species. Reduced metabolic rates, stable environmental conditions, and decreased predation pressure combine to extend lifespans far beyond surface relatives. Some cave fish live over 20 years, compared to typical lifespans of 3 to 5 years for related surface species. Convergent evolution has produced remarkably similar adaptations in geographically isolated cave systems. Cave fish from different continents have independently evolved similar body shapes, sensory systems, and behavioral patterns. This convergence suggests that cave environments create such specific selective pressures that evolutionary solutions become predictable. Size. Modifications occur frequently in cave environments, with species often becoming either much larger or much smaller than surface relatives. Limited food resources favor smaller body sizes in some lineages, while absence of predation pressure allows other species to achieve sizes impossible in surface environments. The extreme specialization of cave-adapted animals makes them among Earth's most vulnerable species. Environmental changes that would barely affect surface species can drive cave species to extinction, making these underground ecosystems critical conservation priorities as climate change alters groundwater systems and surface hydrology. In surface jungle ecosystems, energy flows follow predictable pathways from sunlight through photosynthetic plants to herbivores and predators. Underground, this fundamental relationship transforms into something far more complex and mysterious. Energy, webs that operate according to entirely different principles. Detrital input forms the foundation of cave ecosystem energy flow. Organic matter reaches underground systems through multiple pathways. Leaf litter washed into cave entrances during storms, root exudates seeping through soil layers, dissolved organic compounds carried by groundwater, and organic particles transported by underground streams. This continuous rain of surface-derived energy sustains entire underground communities. Decomposer organisms assume roles of primary importance in cave energy webs. Bacterial and fungal communities break down complex organic compounds into simpler molecules that other organisms can utilize. These decomposers don't simply recycle nutrients, they actively transform them into forms specifically adapted for cave ecosystem requirements. Detritivorous invertebrates represent the next trophic level in cave food webs. Millipedes, terrestrial. Isopods and various arthropods consume decomposing organic matter, while simultaneously fragmenting it into smaller particles. This physical breakdown dramatically increases surface area available for microbial colonization, accelerating decomposition rates and nutrient release. Guano deposition by bats creates nutrient hotspots within cave systems. Bat colonies can deposit several tons of guano annually in large cave chambers. This concentrated organic matter supports explosive population growth of specialized invertebrates that feed exclusively on guano deposits. Some cave ecosystems derive over 70% of their total energy input from bat guano. Predatory. Relationships in cave systems often involve multiple trophic levels compressed into simplified food webs. Cave-adapted spiders, salamanders, and fish typically function as top predators, but their prey base consists primarily of detritivores rather than herbivores. This compression eliminates traditional primary consumer roles and creates unusually direct connections between decomposers and higher-level predators. Nutrient cycling rates in cave ecosystems vary dramatically based on environmental conditions. In areas with consistent water flow and moderate temperatures, decomposition proceeds rapidly. However, in chambers with limited air circulation or extreme chemical conditions, organic matter can persist for decades without significant decomposition. Dissolved. Organic carbon transport creates invisible energy highways throughout cave systems. Groundwater carries complex organic molecules over distances of several kilometers underground. These mobile nutrients support microbial communities in areas that receive no direct detrital input, extending ecosystem boundaries far beyond visible. Cave passages. Symbiotic relationships enhance energy efficiency in resource-limited cave environments. Many cave animals host specialized bacteria that help digest complex organic compounds or provide essential nutrients unavailable from primary food sources. These partnerships can increase energy extraction efficiency by 20 to 30% compared to non-symbiotic relatives. Seasonal variation in energy input reflects surface ecosystem dynamics. Tropical wet seasons increase organic matter transport into caves, while dry periods reduce external energy sources. Cave organisms have evolved strategies to exploit brief periods of resource abundance while surviving extended periods of scarcity. Biomass distribution in cave ecosystems differs dramatically from surface communities. Microbial biomass often exceeds animal biomass by factors of 10 to 100, reversing typical ecosystem patterns. This microbial dominance reflects both the importance of decomposer communities and the energy limitations that constrain higher trophic levels. Energy. Storage mechanisms become crucial in unpredictable cave environments. Many cave animals accumulate large fat reserves during favorable periods. Some species can store energy equivalent to 40% of their body weight, allowing survival through extended periods without food input. Cannibalism and necrophagy represent important energy recycling mechanisms in cave ecosystems. Limited external energy input makes recycling of organic matter within the system critically important. Dead organisms provide concentrated nutrient sources that support scavenging species and bacterial decomposer communities. Chemical energy sources supplement detrital energy in some cave systems. Hydrogen sulfide seepage, methane deposits, and mineral precipitation can support chemosynthetic primary production that operates independently of surface energy input. These alternative energy sources can support substantial biomass in caves with limited detrital input. Energy flow efficiency in cave ecosystems often exceeds that of surface communities. Limited energy availability creates strong selective pressure for efficient resource utilization. Transfer efficiencies between trophic levels can reach 20 to 25%, compared to typical values of 10 to 15% in surface ecosystems. The temporal stability of cave energy webs contrasts sharply with the seasonal fluctuations, characteristic of surface ecosystems, creating unique evolutionary pressures that favor efficiency over competitive dominance. Subterranean jungle ecosystems have emerged as natural laboratories that are revolutionizing multiple fields of scientific research. These underground environments provide unique opportunities to study evolutionary processes, extreme biology, and ecosystem functioning under conditions impossible to replicate in traditional laboratory settings. Environmental DNA sequencing has transformed our understanding of cave biodiversity. This technique allows researchers to identify organisms present in cave systems without direct observation by analyzing genetic material extracted from water and sediment samples. Recent eDNA studies have revealed that cave ecosystems contain 3 to 5 times more species than previously documented through traditional survey methods. Evolutionary biology research in caves provides direct evidence of speciation processes operating over relatively short time scales. Island. Populations of cave-dwelling organisms offer natural experiments in evolutionary adaptation. The Puerto Rican cave coqui frog, for example, has diverged into multiple distinct populations within individual cave systems over periods of less than 10,000 years. Biogeochemical cycling research in cave environments reveals previously unknown pathways for carbon, nitrogen, and sulfur processing. Cave bacterial communities perform chemical transformations that don't occur in surface environments, including novel forms of nitrogen fixation that operate under anaerobic conditions. These discoveries have implications for understanding global nutrient cycles. Astrobiology research increasingly focuses on cave ecosystems as analogues for potential extraterrestrial environments. The metabolic pathways discovered in cave organisms provide models for how life might exist in subsurface environments on Mars, Europa, or Enceladus. NASA has established research programs specifically focused on cave ecosystem studies. As preparation for future space exploration missions, biotechnology applications derived from cave organism research show tremendous promise. Enzymes produced by cave bacteria operate under extreme pH and temperature conditions, making them valuable for industrial processes. Several pharmaceutical compounds derived from cave microorganisms are currently undergoing clinical trials for Cancer and infectious disease treatment. Climate change research utilizes cave ecosystems as sensitive indicators of environmental shifts. Cave temperatures and hydrology respond to surface climate changes with precise measurability. Long-term monitoring programs in tropical caves provide data on regional climate trends that extend back thousands of years through analysis of speleothem. Growth patterns. Advanced imaging technologies have revolutionized cave ecosystem documentation. High-resolution underwater cameras equipped with specialized lighting systems can document cave organisms without disturbing their natural behaviors. Thermal imaging reveals previously invisible patterns of biological activity and water flow. In complete darkness. Genetic sequencing projects focused on cave organisms have discovered unprecedented levels of genomic adaptation. Whole genome analyses reveal that cave-adapted species have undergone extensive genetic modifications, affecting metabolism, sensory systems, and developmental processes. These genomic changes occur much more rapidly than predicted by traditional evolutionary models. Ecological modeling research uses cave ecosystems to test theories about community assembly and species interactions under resource-limited conditions. The simplified food webs and predictable environmental conditions in caves allow researchers to test ecological hypotheses with greater experimental control than possible in complex surface ecosystems. Microbiome research in cave animals reveals symbiotic relationships of extraordinary complexity. Cave-dwelling organisms host bacterial communities that perform essential functions, including nutrient synthesis, immune system support, and detoxification. These symbioses represent some of the most intimate interspecies partnerships documented in nature. Conservation biology research in caves addresses the extreme vulnerability of cave-adapted species to environmental disturbances. Studies document how slight changes in surface land use, groundwater chemistry, or temperature can cause cave species extinctions. This research provides critical data for developing protection strategies for cave ecosystems. Biomineralization research examines how cave organisms interact with mineral precipitation processes. Some cave bacteria actively control crystal formation, creating unique mineral structures that don't occur in purely chemical systems. Understanding these biological mineralization processes has applications for materials, science, and nanotechnology. Comparative phylogenetics research traces the evolutionary relationships between cave species and their surface relatives. Molecular clock analyses reveal the timing of cave colonization events and the rates of evolutionary change in cave environments. These studies show that cave adaptation can occur much more rapidly than previously believed. Ecosystem engineering. Research documents how cave organisms modify their physical environment through biological processes. Bacterial biofilms alter water chemistry and rock dissolution rates, while animal activities create new habitat structures. These ecosystem engineering effects can persist for decades and influence community development patterns. Current research. Frontiers include investigation of cave ecosystem responses to climate change, development of non-invasive monitoring technologies, and exploration of previously unknown cave systems using advanced geophysical techniques. As we stand at the threshold of unprecedented technological capabilities for exploring Earth's most remote environments, subterranean jungle ecosystems represent both urgent conservation priorities and windows into possibilities for life throughout the universe. These hidden worlds face mounting threats while simultaneously offering insights that could reshape our understanding of biology, evolution, and the potential for life beyond Earth. Conservation challenges for cave ecosystems extend far beyond traditional protected area approaches. Surface activities occurring kilometers away from cave entrances can fundamentally alter underground environments through changes in groundwater chemistry, hydrology, and nutrient input patterns. Agricultural runoff containing pesticides and fertilizers can eliminate entire cave communities within months of first exposure. Climate change poses existential threats to cave-adapted species through mechanisms that remain poorly understood. Temperature increases of just 2 degrees Celsius can exceed the thermal tolerance limits of highly specialized cave organisms. Altered precipitation patterns affect groundwater levels and underground stream flows that many cave species require for reproduction and feeding. Habitat fragmentation represents a particularly severe threat to cave ecosystems. Many cave systems exist as networks of interconnected chambers and passages. When surface development disrupts groundwater connections between cave segments, previously continuous populations become isolated. The small population sizes typical of cave species make them extremely vulnerable to local extinctions. Tourism pressure on accessible cave systems creates complex management challenges. While cave tourism can provide economic incentives for conservation, human visitation introduces foreign microorganisms, alters atmospheric conditions, and can cause physical damage to fragile cave formations. Sustainable cave tourism requires sophisticated management protocols that balance conservation with educational opportunities. Pollution from surface sources presents invisible but devastating threats to cave ecosystems. Contaminants dissolve into groundwater and concentrate in underground systems, where they persist far longer than in surface environments. Heavy metals, organic pollutants, and pharmaceutical compounds have been detected in cave systems located hundreds of kilometers from their original sources. Conservation strategies for cave ecosystems require landscape-scale approaches that protect entire watershed areas. Traditional species-focused conservation methods prove inadequate for ecosystems where species interactions span multiple trophic levels and depend on complex geochemical processes. Effective cave conservation must address surface land use practices, groundwater management, and regional development planning. International cooperation becomes essential for cave conservation because many cave systems cross national boundaries through underground connections. Groundwater contamination in one country can eliminate cave species in neighboring nations. Several international agreements now recognize the transboundary nature of groundwater ecosystems and establish cooperative management frameworks. Astrobiology research applications derived from cave ecosystem studies are expanding rapidly. The metabolic pathways discovered in cave organisms provide blueprints for how life might exist in subsurface environments throughout the solar system. Europa's subsurface ocean, Mars' underground water systems, and Enceladus's hydrothermal vents, all present conditions analogous to terrestrial cave environments. Future exploration technologies will revolutionize cave ecosystem research within the next decade. Autonomous underwater vehicles specifically designed for cave navigation will allow exploration of flooded passages previously inaccessible to human researchers. Environmental sensors capable of real-time monitoring of multiple chemical parameters will provide continuous data on ecosystem functioning. Genetic rescue programs may become necessary for highly endangered cave species with extremely small population sizes. Advanced reproductive technologies, including artificial insemination and embryo transfer, could help maintain genetic diversity in critically threatened cave populations. However, these interventions require detailed knowledge of species biology that currently exists for very few cave organisms. Restoration ecology applications in cave systems present unique challenges and opportunities. Unlike surface ecosystem restoration, cave restoration must address geochemical processes and microbial community structure before higher-level organisms can be successfully reintroduced. Successful cave restoration projects could provide models for ecosystem restoration in other extreme environments. Citizen science initiatives are beginning to contribute valuable data for cave conservation efforts. Trained volunteer researchers can monitor easily accessible cave systems and report changes in species abundance or environmental conditions. These programs multiply the monitoring capacity of professional researchers while building public awareness of cave ecosystem conservation needs. The discovery rate for new cave species continues to accelerate as exploration techniques improve and previously inaccessible systems become available for study. Scientists estimate that over 90% of cave-dwelling species remain undiscovered, suggesting that thousands of new species await documentation in tropical cave systems worldwide. Education and outreach programs face unique challenges in building public support for cave ecosystem conservation. The hidden nature of these environments makes them difficult to experience directly, requiring innovative approaches to help people understand their significance. Virtual reality technologies may provide new opportunities for immersive cave ecosystem education. Looking toward the future, subterranean jungle ecosystems will likely play increasingly important roles in understanding how life adapts to extreme conditions, how ecosystems function under energy limitation, and how biological processes might operate in extraterrestrial environments. Protecting these irreplaceable natural laboratories represents one of conservation biology's most urgent and challenging frontiers.